Thank you, Betty, for your words and explanation about my life. So, thank you for being here for all of you. And I will be presenting some advances of my dissertation work that is not ready yet, but I have some results I want to share. So the title is Land Use Change and Biodiversity, Understanding the Patterns, Driving Mechanisms, and the Impacts of the Mitigation Action. So just to put you in context, in developing countries, we have governments and people trying to fill these basic needs that we have. For example, access to clean water, food resources, transportation, healthcare. Especially in developing countries, we see our resources, natural resources, as a way to achieve this economic wealth. So we look at the resources by the window of development. Specifically at Peru, that is located in South America, we have most of our territory as Amazon forest. And as you can see in the light green color, a big part of this territory is protected. But also, what happened exactly outside the protected areas? So the government and also the people is using the forest for generating resources, economic resources. And unfortunately, the land use change that are taking place in Peru is conducting to forest loss. So this is an image of Peru, and it's showing the hotspots of deforestation in the territory, and specifically in the Amazon, for 2017. So deforestation is a really important factor that is influencing biodiversity, and it's kind of well understood. However, what happened after the deforestation process takes place is not that well understood. So I have two main motivations for my dissertation work. The first one is to answer if the mitigation measures are working or not, and I will be talking about a specific example of riparian forest strips, and also I consider there is a poor understanding of the impacts of different land use types on biodiversity in this kind of human-modified landscapes. And for my dissertation, I selected the bats as a study group because they have several characteristics that make them a really good group for ecological studies. So to start, they have different morphologies and habits, as you can see in the picture, different colors, sizes, and shape of the wings. They provide several ecosystem services. So in the case of the frugivores, they eat the fruits, they disperse the seeds, helping to the forest regeneration. Nectarivores pollinate the plants. Some insectivore species feeds on or eat insects that are considered as plagues. And also other species of bats feed on other mammals, including bats, birds, fish, and frogs. Other characteristic is that they are really susceptible to the perturbations in the habitat. So all these characteristics makes them a really good group as an indicator of disturbance in the landscape. And in the case of Peru, we have 180 species of bats. So just to start, what happened when in an area that used to be continuous forest, we end up having only these riparian forest strips? So imagine you have this beautiful forest, then people decide to take down the major part of the forest, but they keep this riparian forest strip because under the law, under the Peruvian law, we need to keep this strip of 25 meters of forest to each side of any water sources. So for the wildlife, what this will represent? Will they prefer to stay just in the area that maintains the forest, or they will take the risk and move across the clear area? So for answering this part of the dissertation, I moved in 2014 to San Martin, that is in the north-central jungle of Peru. And it's a department that has a really high rate of deforestation. But also it's a department that has a really good territorial zonation that is not replicated in other departments. And they actually encourage this kind of measure for mitigation, like maintaining the riparian forest strips. So I had the chance to collaborate with a private company, and they were planning to establish an oil palm plantation in this part of San Martin. So they had these 1,300 hectares that they cleared almost completely in 2014, for which year I don't have an image. But you can see how the territory ended up in 2015. And they use for that the heavy machinery. So for this part of the dissertation, I had two main objectives. The first one was to evaluate the impacts of the maintenance of these riparian forest strips on bat diversity. in this human-modified landscape, and also to define if there was an effect of time on the biodiversity. And this is the design I use. So I established 3 different treatments. And I want you to pay attention because I'm gonna be mentioning these treatments a lot of time after this. So the first one consisted in continuous forest without the presence of any kind of water source, just forest. The second one had the presence of streams, and they were surrounded by continuous forest. And the third one was the area that the company already cleared. So they have the riparian forestry that had 25 meters of forest to each of the sites, and they have the clear area in there. And for each of these treatments, I had 2 sites. So this is how it looked in reality, the riparian forest strip surrounded by clear area. We had the forest at the riparian strip, then the edge that was really sharp, and the matrix, what I consider the matrix that was clear. But as you can see, the trees that they cut down were still there on the ground. And this is how I sample bats. So I use a mist net that looks kind of like this. Bats get attached in there and we have to release them. So we established 3 different transects with 2 points where we located the mist nets in L shape. So we have 1 transect on the side of the stream, 25 meters away at the edge we have another transect, and 100 meters away in what I consider the matrix, we had the other transect with 4 mist nets. In total, we distribute 12 mist nets in this setting. And this kind of design was replicated for the other treatments. So when we had the stream surrounded by forest, I established a transect on the side of the stream, and then in what was considered as edge in the other treatment, here is forest, because we didn't have a clear area. And also 100 meters away, another forest transect. And the same for the forest, the continuous forest treatment. So we worked there since dusk to midnight, around 6 hours, and we were capturing bats with the nets, checking the nets, releasing the individuals, measuring the individuals, and using different guides to identify the species. So we end up having 48 nights of sampling, and we captured 1,323 individuals that belong to 49 species, and they belong also to 5 different families. But you can see in this pie chart that most of the species belong to the Phyllostomid family. And this is because the mist net is a methodology that allows us to capture bats from that family mainly, because the other species from the other families avoid the mist net, they detect the mist net easily. All the species belong to 7 trophic gills. So the first objective, again, that was evaluate the impact of the presence on these riparian forest strips. Just to show you that the sampling effort that we had was enough to have a good representation of the Phyllostomid bats. As you see, the accumulation curves are reaching the asymptote or the plateau. But when we took a look into each of the treatments that have different habitats, we see that in this case, rarefaction curves are showing that there is not a significant difference among the richness that we register in those habitats. But if you see the darkest line that corresponds to the edge, if we will continue with the sampling, maybe the number of the species will differ from the other habitats. And in the case of the stream surrounded by forest treatment, and both habitats were not different in the amount of species registered. I also took a measurement of the species richness using the Chow estimator, that is a non-parametric index. And with this calculation, we see that only the edges at the riparian forest strips surrounded by clear area treatment is showing a difference that is significant. And when we took a look to the abundance of a species, I use this rank abundance curve. So in this graph, you have in the y-axis the capture rate, and in the x-axis the species rank. The species that are located at the position 1 are the species that have the highest abundance, and the species that are on this part of the axis are the ones that have the lowest abundance. So if you take a look to the shape of the riparian forest strips surrounded by clear area treatment, you see that there is a dominance of two or more species, but that doesn't happen in the forest treatment. And you have as an intermediate curve, the one for the streams surrounded by forest. So actually, it appears that the heterogeneity in the habitats in this treatment is driving the difference among them. So But that was for the treatments, entire treatments, without considering the difference in the habitat. I replicate or did again the rank abundance curve for each of the different habitats inside or within each of the treatments. And you can see here that at the stream surrounded by forest treatment, the shape of the curves and the kind of number of the species that were dominant is not that different between habitats. But if you take a look into the riparian forest surrounded by clear area treatments, at the stream and at the edge, we have a dominance of a couple of species. That is not what happens at the matrix, that is the clear cut. And here, what I'm doing is analyzing how different were the species composition of the assemblages in these treatments, using the non-metric multidimensional scaling analysis. So you see that the x-axis is separating the sites from the treatment that didn't have the presence of any water sources from the sites and treatments that have the presence of streams. And in here, what I'm doing is showing all the transects together that I evaluated. And what it's showing is that the x-axis, the first axis, I'm sorry, again, is separating those habitats that only had forest and not water sources from the ones that have the presence of the streams or influenced by them, like the edge of the riparian forest strips, and from the areas that were matrix with clear cut. So when we focus in the second objective, that was to see if there was an effect of the time on biodiversity, I only pay attention to this treatment. And for that one, I have one side that was recent clearing area, because when we sampled the bats, the area was cleared just a month before. But the other side, the second one had an area that was clear in 2002 and an area that also was clear in 2010. And actually in the matrix, or what I consider the matrix, and I put the mist nets, was clear in 2014, but at least 2 months before we started the sampling. So again, I'm showing here the rank abundance curve, just to make a comparison between each of the habitats in these two sites, the recent clearing site and the old clearing site. So you can see that at the stream level and at the edge level, the recent clearing site has a dominance of the Phyllostomous status species. That doesn't happen in the matrix habitat when you compare with the old clearing site. And also other characteristic that I can see here is that the recent clearing site is showing a bigger number of rare species at the stream level and at the matrix level when it's compared with the old clearing site. So just to remind a little bit about the conclusions that we're making in terms of these results is that the heterogeneity in the habitat is driving the species diversity among the treatments. The bat assemblage is separated by the habitat characteristics. The bat in the riparian forest strips are dominated by a couple of species and also have several rare species. And the difference between the recent clearing area and the old clearing area is mainly driven by the presence of Phyllostomous status and also by the amount of rare species. So this might give us a little bit of information on how important this riparian forest strip can be, because they are maybe maintaining the ecosystem services and also serving as refuges and corridors for the bat species. But are we sure that this matrix is going to stay clear forever and ever? Or what happens if people decide to, for example, make this palm plantation to grow, or maybe they decide to establish a crop of any kind, or they abandon the land and the land starts regenerating naturally with some fast-growing species of trees, or maybe if they abandon it, and then they decide to put some cattle there and they maintain this pasture land. So is the wildlife going to perceive the landscape in the same way as they were doing when the matrix was clear or not? So what happens specifically if that matrix is used for a plantation and for cattle pasture? So for this part, I moved south to Peru, in Madre de Dios. In 2016, I was there during 5 months, and last year during 2 months. And Madre de Dios is considered as the capital of biodiversity by the Peruvian government. And actually, if you see in the light green color, most of the territory of the department is protected. However, we have this highway that connects Madre de Dios with Brazil. And it's the focus of deforestation due to different activities, especially the presence of gold mining, illegal gold mining, but also several agricultural activities. So I focus in two, as I mentioned, two different production activities. Cattle pasture that has been in the area since the 1960s, and the papaya plantations that has been, we have like a boom, an explosion of papaya plantations in Madre de Dios since 2013. So in this part, I have two objectives. First, to describe what is happening, what are the patterns of biodiversity depending on the agricultural land that is taking place in the area, and having a little bit of focus on what happens at the forest edge. And the second one is to understand a little bit more about the mechanisms that are underlying the patterns. So I located 12 different sampling sites following the interoceanic highway. 6 of them corresponded to papaya plantation and 6 of them to cattle pasture. They were separated at least 4 kilometers to ensure independence, and I cover a linear distance of 170 kilometers. I will be showing just the data from 2016 in here. So I have this design that is kind of similar to the previous one, but in this case, I was sampling 3 different habitats: the forest interior, the forest edge that was normally sharp, as you can see in the picture on the top, and the production land that was or papaya plantation or cattle pasture. In each of these habitats, I place a transect that, in the case of forest interior and production land use, consisted in 5 different points separated by 17 meters that had each of the points a pair of nets in L shape. But at the forest edge, because I wanted to cover the 3 microhabitats, I located 15 mist nets in triplets in T shape. So I have 1 mist net going inside the forest, 1 parallel to the edge to catch the animals that were going out and inside the forest, and 1 going to the production land use. So once again, we were working during the night, capturing bats from dusk to midnight, and we spent 1 night per transect during each of the visits, and we did 2 visits in 2016. So in this time, we captured 3,355 individuals that belong to 54 species, and they also were distributed in 6 families. And once again, you can see that in the pie chart, the Phyllostomide was the family that presented the highest amount of species. So just to focus, now we have to focus on Phyllostomide because they were the better sample family. So in here, what I want to show you is that in terms of species richness and capture rate, when comparing the different habitats at the different site categories, we see that there is a tendency to have, for the papaya sites, to have a higher richness and also kind of a higher capture rate, especially if you focus on the production land habitat. And this is really important because we have much more captures in the papaya land. And this is mainly driven, as a comment from the field, that we were capturing a lot of frugivore bats from the genus Artiveus that were feeding like crazy on papaya. They really like papaya. And this image is just to show you how each of the species that we captured along the whole study were present or absent in each of the habitat that we sampled. So if you focus on the first position, that frugivore species was present on the forest, at the edge, at the papaya, and at the cattle pasture. But if you focus on the third position, that was another frugivore species, was again present at the forest, edge, papaya, but it's blank there because it was absent in the cattle pasture. So something important to notice here is that cattle pasture showed the highest amount of species that were absent, and actually an entire trophic yield was lost in there. The green insectivore that is considered as a group that is indicator of perturbation. Another thing to highlight is, for example, the presence of this carnivore species. That was the only species of carnivore bats that were present in the study, and only was captured in the forest. And this makes sense because it's a species that is normally related to mature forests. So looking at what happened with the assemblage composition, you can see that at the papaya sites and pasture sites, the composition of the species among habitats were different, but the difference is stronger at the pasture sites. And when comparing forest in the papaya sites and cattle pasture sites, I wasn't expecting them to be different in terms of the composition because they all were forest, and this is what we found. But at the edge, I was expecting the papaya to have, or the cattle pasture to influence the composition of the species of bats at the edges. But with these results is not showing that there were a difference in their composition. And in terms of the agricultural land, there is a tendency for the cattle pasture habitat to be different in composition from the papaya habitat. So general conclusions, on average, papaya seems to be better for bats than cattle pasture. One entire trophic yield was lost in the cattle pasture, and the habitat is influencing the bat species composition. But what happened with the mechanism? Because if you remember, the second objective was to try to understand a little bit more about the mechanisms that are driving these patterns. So we have to consider that there are several factors that are acting to determine what's going to be the patterns that we observe. And those factors are at the large scale factor, for example, the forest cover surrounding each of my sites, but also at the local level, you can have vegetation structure or, for example, temperature. So imagine you're a bat, a beautiful bat, and you're flying by the forest, and what would you prefer? Vegetation that is as dense as this one that won't allow you to move freely or fly freely, or maybe this kind of density of vegetation. So I decided to measure vegetation density in different ways. I started estimating the large tree densities, meaning those trees that have more than 10 centimeters of DBH. And I found that the highest density was located at the edges instead of the forest interior. We also measured the tree density for the smallest trees that have less than 10 centimeters DBH and for the vines density. And we didn't find any patterns of difference between edges and forest. We also take a measurement of the vertical profile of density for the vegetation using this imaginary cylinder methodology. And we were trying to have a measurement of how dense was the vegetation horizontally. Thinking again as a bat, it's important to know how many obstacles they need to fly by when they fly. So we took these pictures using this white screen, and I was always the photographer. I was located 5 meters away from the screen, and we end up having these kind of pictures that I should be transforming into white and black. And the idea is to estimate the amount of percentage of black color in there that will represent the density of vegetation. It's like if you will be estimating forest and non-forest in a map. In addition, we also calculated or estimated the canopy height, and we found that the canopy height was higher at the forest interior than at the edges. And we took measurements from temperature during all the study, during each of the nights that we sampled bats at the forest edge and at the production land. And for the large scale, we have this estimation of the forest cover using a center point each of our sampling sites, and we were using different radius. For example, this image is for the 5-kilometer radius of one of my sites. So the idea is that I have all or maybe part of the explanatory variables that I measure, and together with my response variable that will come from the patterns I observe, it can be abundance, general abundance, abundance of each of the species, the evenness of maybe similarity between the assemblages. I can create some models that will help me or help us to understand a little bit more about the mechanisms that are working behind these patterns. But until now, I have been talking a lot about phyllostomid bats, that they are the most captured with mist nets. But what happened with the other species that belong to the families that were also captured but not well represented with the methodology that I used? For those species, what we did was using these acoustic surveys. So we were using these recorders, bat recorders that have they can detect really high frequencies at which the bats call. And the important thing to know here is that the insectivore species that are mostly recorded by the acoustic surveys have a specific vocal signature that allow us to make the difference between the species. So we were, during the night that we were sampling bats, we were also placing these recorders. And we end up having this huge amount of data, this 160,000 audio files that I'm analyzing. And I estimate that at least I will have 10 additional species. And this figure, what it's trying to do is to show you some examples of the summary of what density, which characteristic frequencies have the higher and lowest density. So in this site that was a papaya site, if we compare to this pasture site, you can see that a papaya habitat there is a high density of one species that is calling at a characteristic frequency that is around 20 kilohertz. But that species that is really apparently really active in papaya is not that active in pasture land. So these kind of summaries give us this important information. So to tell you a little bit about future directions. So we have several captures that were rare species, and some species like this Pteroptera winnii, that is a big winged bat. And the record that we have constitutes a range extension. And you can see that the characteristic for the species that they have these suction caps on the wings and feet that allows them to stay attached to the leaves that they use to roost. That is Heliconia, normally, from the genus Heliconia. And this species was described in 2014 when I was present there, when they captured that individual in the north of Peru, in Loreto. And so this is a really far away new register for the species. And this is another species that is considered as rare. It's a bonneted bat, Junus naurus. And the characteristic for this species is that they have this white fur on the side of the body and under the wing. This is pretty good. And for Peru, we have few registered, and actually the closest one is also in Madre de Dios, in an area that is a little dot or spot in here that correspond to Pampas habitat. And we actually captured this individual in a cattle pasture, kind of similar, but not at the end. So it's a good record for like increasing the amount of information about the natural history for this species. And just because I wanted to show this, and it's really rare and normally it's not captured, this is a Pharonecteris toxopilum, it's a viceroy bat. And you can see that the species has this dimorphism, the females and the males. And it's considered as data deficient by the International Union of Conservation of Nature. And we had 3 of these individuals. It was really exciting to have them. So we can also provide a new information about the habitat where we capture them and contribute to take that data deficient out from the title of this species. And something that came out when we were in the field is the mercury analysis. I mentioned that one of the activities that is really extended in the department is the gold mining, illegal gold mining. So they use mercury to separate the gold from the substrate. And there has been in the last months, actually, several studies that are using bats as an indicator of contamination of mercury. And they were comparing sites that were highly contaminated with sites that were not contaminated. So I had the chance, well, it's not a chance, but we were clipping hair from the back of the bats just to mark them, to avoid having recaptures not considered. And we kept the fur. So in collaboration with Dr. Frederick Labs, we are running this mercury content analysis in all 9 species. And you can see that what we will do with this information is to complement the grading of contamination that is already published in the previous studies. And I took advantage of my presence in the field to try to change a little bit of the bad perception that local people had on bats. And I prepared this kind of posters, in Spanish of course, for local people and for the landowners that allow me to go into their land. And in collaboration with a friend and colleague in Peru, she created these cool cartoons from bats. And the idea is to have a more friendly way to approach kids, to make them think about bats in something that is good and not bad as they think. And I also, since I was in the field, I was able to give some talks in the schools, in the area where I was sampling, in Lima, the capital of Peru, and in Puerto Maldonado, the capital of Madre de Dios. Thank you so much for your attention. I want to thank, first of all, to Betty, because you have been a really important support for me during these whole years, to all the members of my committee, 2 of them are here now, to my lab mates that have been with me during these whole years, the ones that are here and the ones that already graduate, and to all the funding sources. These people, including you, that were with me in the field suffering everything, and I was requiring them to be covered and just show their eyes because I didn't want them to have leishmania. And a special thanks to my dad that is not here anymore, but he was really important for the first part of my thesis. And he was providing me logistical support, emotional support, and in part I'm doing this PhD because of him. So thank you.